Slovenian Education Foundation, ASEF, is an organization that enhances American and Slovenian education activities, uniting Slovenian scholars and educators globally. It has three core values: academic excellence, character formation, and dedicated service, permeated by special care for Slovenian heritage. In the ASEF podcast, we host ASEF professors to get to know their research, best practices from their field, their vision for the development of that field in Slovenia, and how they envision collaborating among Slovenians worldwide. Welcome to another episode of the ASEF podcast. So far, we have been exploring the physics, computer science, and artificial intelligence, as well as cancer research. And today we will be talking with three ASEF professors about biochemistry research. With us are Professor Jernej Murn, an assistant professor at the Department of Biochemistry at the University of California, Riverside, and Professor Boštjan Kobe, a professor at the Department of Chemistry and Molecular Biosciences at the University of Queensland. Thank you all so much for joining us today. First, I would like to ask you if you can briefly introduce yourselves, tell us about where you studied, for how long you've been in the field, and maybe something about your current research.
Thank you, Tanya, for the invite. Thanks for having me. Well, my research career started really relatively late, I should say. I studied pharmacy really because I thought sort of knew I was interested in biology, that there was something to it that I liked, and chemistry, and there were other things like music and so forth. But these were really sort of my fields, and sort of that directed me to pharmacy because I didn't know much else. I didn't know biochemistry really existed. And so I think that set me up on the, this trajectory. And, it took a while before I realized that there is something in between this brute, sort of biology as people have it usually in mind, trees and birds and so forth, and, and chemistry, right? So from there, it took a while before I really discovered this field largely through reading. And from pharmacy, so I graduated there at our Faculty of Pharmacy in Ljubljana, and from there I moved on to Paris. There was an opportunity to study at the lab of functional genomics. There I got in touch more with the molecular biology. I saw that genes were expressed and so forth, and that proteins existed that one could actually study. So that was really late for me. I was in my 20s, mid-20s and so forth. But then when I got a bit more into it, I sought additional opportunities. And so I moved on to do a postdoc at Cold Spring Harbor Lab, where I got sort of pushed a little bit back into not really molecular biology, more into cancer. And I didn't quite find myself. So, so then this really, but this experience solidified my view, and sort of there I really decided what I was interested in, that it was really molecular biology. And so that got me to move on to Boston, to Harvard, where I did a pretty long postdoc. I think it was close to 7 years or so. I got into a lab of Yang Shi, who is an epigeneticist. So we studied histone modifications, or rather other people in the lab did. And I was an exception there because I stumbled upon a protein that happened to be an RNA binding protein. And so that set me up on this track of RNA biology. It wasn't before, let's say, perhaps 10 years ago that I really started thinking and working on RNA. This stayed with me. And so the last 4 or so years since I've had my lab at the University of California here in Riverside, this is our going. So we are really exploring very basic RNA biology, coding and non-coding RNA, how it's processed, how it regulates cellular responses, cellular decisions, and how it may be misregulated in in human disease. So in a nutshell, that's my trajectory.
Okay, thank you very much.
So basically, I studied chemistry in Ljubljana. I was interested in chemistry, and I think at the time as an undergraduate student, I was interested in inorganic chemistry, I think most. But then towards the end, so I did a research project as part of my diploma work, which was determining a crystal structure of an organometallic molecule with under the supervision of Van Leeuwen. And I fell in love with structure, crystallography and other methods, but structure of chemicals and molecules, and realized that people are actually determining structures of macromolecules, proteins, and nucleic acids, and so on. So, and I decided that's what I wanted to do and continue further in my career. And this was kind of one challenge in my career, was that at the time, really, in Slovenia, there was no lab, no equipment to do macromolecular crystallography or structural biology. That's why I ended up in the US. It was kind of in a way more streamlined to get a PhD, to go to do a PhD in US than in most European countries. So that's kind of how I ended up in the US. So I did my PhD in Dallas, Texas, and that's where I worked on a protein called RNase inhibitor, on the structure of that protein. So there is a little bit of connection of RNA. So this protein was basically an inhibitor of enzymes which cleave RNA. And it did also then determine a little bit what I continued to do after that. So after I finished my PhD, I stayed a little bit as a postdoc in the US, but I decided I wanted to change. I didn't want to live forever in the US. I just didn't like, kind of from the cultural point of view, from work where I was, was absolutely great, but and I was kind of at the position in my life where I kind of wanted to experience something different. So I got a job in Australia, and I've never previously visited Australia. Just thought I'd try something different. Thought I'm gonna go there for a couple of years and then go back to Europe. But then obviously things worked a bit differently, and I then stayed in Australia ever since. So I first started in Melbourne. I started my own group there, and then I moved to Brisbane at the University of Queensland. So all this time, we continue to do structural biology. So that's kind of what I do. We'll look at 3D structures of macromolecules, in particular proteins and protein complexes. The reason we do this is because we want to understand how these proteins work. So proteins obviously fold up in a particular complicated structure, and it's basically that structure that determines what they actually do in the end. So we try to understand function or molecular aspects of function from looking at the structure. And in the recent period, we've really been focusing in particular on proteins involved in the innate immunity response. So this is part of the immune response in humans that is conserved across all organisms pretty much. And it's been keeping us busy for a while.
Okay, thank you very much for this very nice introduction of yourselves. So now let's maybe kick things off with some basics, even though Professor Kobe already gave us a little bit of an introduction into proteins. Maybe let's talk a little bit more about what are DNA, RNA, and proteins. And maybe Professor Murn can tell us a bit more about DNA and RNA, and then Professor Kobe about proteins.
DNA, RNA, and proteins, these are all polymers. They are biopolymers. And they're really the carriers of life. So life revolves around these three types of molecules. And they've been connected, through the so-called central dogma that was put together through decades of research, but really, then written down first by Francis Crick in mid-60s. And, if we go one by one, DNA is really as you may know, right, the blueprint of life. This is where the information is stored. So it's like a hard currency. At least life on Earth is all needs some medium to store information, right? It need not necessarily be DNA, right? Certain viruses have RNA as their genome, as their blueprint. But DNA sort of has established itself as a bank, a pretty stable molecule that can then be replicated, and it can give rise, it can mobilize the information. And this mobilization is carried through by RNA. right, which serves both as a messenger of information that can then in turn be translated to proteins, as we know, but it can also serve many other roles. So it's a very versatile molecule. I'm a bit biased here, obviously, right? But that's, I'm just telling you the facts. I'm not hopefully saying something wrong. And so there are many types of RNA. It's a mobile molecule. It's it can do things. We can chat more about that. But so if you're talking about the central dogma, this is really what encodes proteins. So it serves as a template for translation of proteins.
Yeah, so basically proteins are what is then encoded by RNA and translated into proteins. So proteins are polymers, but unlike nucleic acids, polymers, they're polymers of amino acids. In humans, we use 20 different amino acids, and it turns out, you can make, obviously, almost infinite number of combinations of these 20 amino acids, and that gives us kind of different polymers that actually end up then folding up in different ways. And it's really this how they fold up, so that in the end of the day determines what they actually do. And that's why what I'm really interested in is looking at that three-dimensional structure and understanding how do these different structures lead to different functions. And I think proteins, they probably do most of the things around the cell. Obviously we heard a lot about the different things, it's particularly RNA, but also DNA do functionally. But at the end of the day, I think proteins do a lot of things, or most things inside the cell. And so some of them are located in or different proteins are located in different environments. For example, we've heard about the nucleus, heard about the cytoplasm, so that's all inside the cell. There's also proteins that get exported outside the cell and they function there. There's proteins that sit in the membrane and kind of function in this communication in and out of the cell, or even between cells, and so on. So proteins have functions like enzymes, so they can catalyze chemical reactions. That's probably one of the best known functions. But others also have structural roles, or they are involved in kind of signaling pathways in the cell to tell the cell what to do, or what needs to happen at certain times in the cell. So so that's I think in a nutshell what proteins are.
Okay, thank you very much. Okay, so Professor Murn, you were mentioning central dogma, and if I understand correctly, this is when DNA is like, so DNA makes RNA and RNA makes then proteins. This would be like
A simplified view of
yes,
it.
a very simplified view, exactly. But not too long ago, you had a lecture for ASEF fellows, and you mentioned during that lecture the RNA world hypothesis, where RNA would exist before DNA. Do you maybe want to tell us a bit more about the RNA world hypothesis?
Well, sure, yeah, happy to. So this hypothesis goes back to the fact that we have three different types of polymers, biopolymers, right? And so after the Earth cooled down or started to cool down, life had to emerge, as we know, somehow. And it's unlikely that all three types of molecules co-emerged and that they would be connected in this sequence, right? DNA goes to RNA goes to protein. And so, given the conditions that are thought to have existed on Earth back then, 4 billion years ago or so, it seemed that RNA through this is experimental work, right? That people have tried and are still trying to work out, right? What could really have happened back then is that RNA is the only chemically the only molecule that could have emerged and carries the capacity that would allow it to to carry information, to propagate itself, and also do a little bit, right? Do some enzymatic facilitate enzymatic reactions. It's a hypothesis, so it's untested, and we'll probably never know if it's a good hypothesis, right? But it seems like, compared to DNA and proteins, that this is as close as we could get to having something that could, in theory, have started life. It may not have been RNA as we know it today. It could be some other form of RNA, some different backbone, etc., a little different chemistry. But in principle, this is probably the best explanation that we currently have as to how life started. And so it's an RNA world hypothesis. So back then, from that prebiotic soup, perhaps RNA emerged. Again, I'm biased here, and I sort of support that. I don't know if others have any other thoughts on that.
Well, I can add something. I mean, I guess it's one of these philosophical questions which we'll probably never be able to prove or disprove. But I think there's a couple of things that really I think support this RNA world hypothesis, and one is the ribosome, right? I mean, this is the big complex that makes proteins from RNA, and this complex is made mostly from RNA. And if you go, up the evolutionary tree, more proteins were added, but the core is really made of RNA. So clearly, it was RNA that supported this this function to start with. And the second is, that RNA can have enzymatic functions. I think the ribozymes were mentioned. So RNA is capable of catalyzing reactions, and I think these are two things that very much support this RNA world hypothesis.
Okay, and now turning a bit away from the philosophical questions from how life emerged, more towards the current situation of pandemic and the mRNA vaccinations. So since we are still in the times of the pandemic and there is so much talk about the mRNA vaccinations, and we hear so many different pieces of information about what is the principle of action of these vaccinations. Would you please explain to us what exactly is the principle of action, so we can clear the air once and for all in some more simple terms?
Well, if I start, right, so principle of mRNA vaccinations is really to as opposed to, let's say, the old protein-based vaccines, the versions that have been around for a long time, is to use RNA as a vehicle, as a medium, right, as a messenger that will churn out protein in our cells. So instead of having to work out how to make the protein, how to make it stable, what type of protein, etc., right? This is a very laborious process because proteins are much more complex. They have structure, they are, modified, etc. RNA is much simpler. And so the idea behind mRNA vaccine is to sort of just introduce an mRNA into the body with a purpose to encode a certain epitope, if you can identify one, that will elicit an immune response. And so this way you sort of bypass all the cumbersome steps of developing a protein-based vaccine that better known that have been around for many decades, and also speed up the development of different vaccines. Now the main challenge here is how to deliver mRNA into cells, into the targeted cells, and how to make it stable enough for it to not get degraded, etc. And so this has been the main obstacle, really, right, in the field. Otherwise, the idea has been around for many decades and people have worked on it. But it wasn't before COVID, right, really, that this came to the fore. And behind this vaccine, and the reason why this developed so quickly and now everybody's vaccinated, Part of the reason is that, technologies to stabilize RNA and to deliver it, they have been in the development for over a decade, right? Well over a decade. And so it just so happened that it came together. Of course, many people invested heavily into it. And, within a span of just a couple of months, the mRNA that is now used in FDA and approved vaccines, for example, right, was devised. So this is really the major thing. And the nice thing about using mRNA as a vaccine is that it can quickly be modified if need be, right? So if there is another virus, another strain, another unknown foreign pathogen, it's as simple or almost as simple as changing the sequence of bases in RNA to encode a different protein that might then work in a similar manner to elicit an immune response that would then generate antibodies to protect us, and also cell-based immunity is triggered. I probably, forgot many things that should have been said here, but I think this is sort of the general idea and the reason why things work now, right? Or so we hope.
Yes, since you already touched on the next question that I wanted to ask, which is how did this mRNA vaccination came so out of the blue, and it just became ready at the very right moment? Maybe we can then spend a little bit more time on which other diseases this vaccination could be used for. For example, I didn't know that mRNA vaccination could be used for cancer treatment. I think this is quite amazing. Do you think that this will be used in future? Does it have good prospects?
Yeah, I'm the least expert on this topic, but I still have opinions. So, yeah, in fact, I think the first application of mRNA vaccines was to cancer, as a cancer vaccine. And in fact, it only was applied to a infectious kind of against an infectious agent much later. So 2013, I think, was the first for rabies. It was the first trial for rabies. But yeah, I mean, it's basically, there is really, in the principle of how it works, no any difference between how any of these vaccines works. Basically, we need an antigen which will trigger an immune response, and it just comes down to how do we deliver that antigen to our cells so they can start an immune response. If we deliver a protein, then obviously we're delivering antigen directly. Delivering an mRNA, then obviously we have to make the protein first, and so on. So really, I mean, there's no real difference in theory or in principle how this, in the end of the day, what happens. Therefore, there's really no difference also in how any of these different vaccines to what, against what they can be applied. And cancer, cancer cells is one. I think there's other problems with applying it in cancer, which is, it's not a kind of a foreign organism. It's one of our own cells that has kind of gone out of whack, that we need to start targeting. And I think it's more difficult to make it specific to cancer cells versus our normal cells in our body, compared to perhaps against viruses and bacteria.
But given all that, maybe just one word on that. The theory makes sense, right? We know why it works, but still, the fact that it works so well, or that it worked so well, right? This vaccine and also some earlier trials, I think it's still surprising. I think it got most of us and also the developers by surprise. And if you listen to some of the stories, right, from the heroes, right, who are behind developing this, that they openly admitted that they couldn't believe how well this really works. It's because you're introducing something that's fragile, that you don't know where it's going to go. Is it going to hit one cell locally and then none of the rest, right? How is the immune system going to react locally and so forth? But somehow it just works, right? And so, people would say they didn't bother about, they didn't ponder too much about as to how it works and how is that possible. It just, that was just a beautiful thing, right? That it worked. And it's not just, the COVID vaccine. This pertains to, as you mentioned, right? This could be used in cancer treatment, and other chunks of RNA could be used for other purposes as well, right? So also, let's say, you could affect splicing, you could shut down genes and so forth, right? So RNA can do that as well. So it's a great potential in that. I think that this basic principle that it can work, I think that's something that's quite amazing. It amazes, I guess, me.
Staying a bit on the topic of the whole COVID pandemic, for a drug there needs to be a couple of phases of clinical trials, and in the case of the pandemic, the last phase, as I understand, was skipped and it went immediately to the public. So do you have any opinions on that? Like, for the future, do you think that this is something that will never be repeated again? Or do you think that now maybe like this trials will be reconsidered? Or do you think that there will be any changes?
It wasn't fully approved from the outset, right? So things were sort of bypassed a little bit, right? But given the emergency of the situation, right, we were looking at those, the footage from China, people falling down on the street and so forth, right? So I think that really and how the speed with which the virus spread, I think that prompted everyone or the authorities, right, the FDA and other agencies to sort of grant this so-called emergency use authorization, right? It wasn't fully approved right off the bat, right? So it took a while, and but I think it was a unique case, but I think it's now that we know more about what worked, how it worked, I think now we are better ready as a society, right? As a mankind to fend off any future similar infections or diseases, right? And potentially also other disorders. But I think now the timeline for approval of RNA-based either vaccines or therapeutics could potentially be faster, right? Than what was typical of other types, more traditional therapeutics, chemicals, protein-based vaccines, and so forth.
So I would not call it that any of the process was really skipped here, really. I think it was accelerated. So and obviously in the light of the situation and the seriousness of the situation, I think that was the right call, that that things were accelerated, and maybe some of the trials were not done on a scale or for the length that would normally be done. But obviously, this continues to be monitored. Anytime we realize that there's a problem, we react, and that's what happened, for example, with AstraZeneca vaccine and the blood clots. There was immediate kind of reaction to it, and it was reassessed, who should be getting these vaccines, and that's what needs to happen. So I think the only thing that we still need to wait for is kind of a really longer term consequences of which I think will take a few years, and which is really no different from any other vaccines which we introduced in the past. I mean, sometimes we had to wait for a long time to uncover certain things, but looks like, overall, I'm very happy that we were able to react this quickly and get things reasonably under control.
Now, Professor Kobe and Murn, you both worked in lots of different countries. You, Professor Kobe, you were in US, now in Australia, also studied in Ljubljana. What would you say are the differences that you felt in various different places? Maybe how do you think that Slovenian research could improve?
Yeah, one thing I didn't do is ever really work in Slovenia, so it's very hard to judge how things work in Slovenia. I obviously have a lot of talk a lot with researchers from Slovenia and others, so I hear a lot about it, but I don't have first-hand experience really. Well, I think working in the U.S. and in Australia, the U.S. is very driven. People are very driven. They work hard. I think there's a lot of kind of pressure in a way, kind of in the air. At least where I worked, there was a lot of good things about it. Very well equipped, very in the place where I worked, it was also very collaborative and very open. So perfect place to do science in a way. But on the other hand, there was this kind of expectation. I think that you need you work hard and you work long hours. In Australia, I think there's a better balance in a way, but I think people have to be more self-driven. So it's kind of it works well for some people, works not so well for other people. So at the end of the day, I think what it comes down to, and you need kind of diversity. You need, I think you need kind of an environment where you were able to express yourself in when you have different personalities and so I think the best way to do science is in a collaborative way when people work together, where people are when the environment is inclusive and when things are open, when you can openly discuss things and you don't keep things to yourself. So one other thing you obviously need to do good science is funding. And I think this is a problem. It's not just a problem in Australia or in Slovenia. I think it's a problem everywhere. So for some reason, politicians, despite how obvious it is, especially in the last 2 years, how important science is. Basically, I think science is saving us right now from some really bad situation. But I can't see that there's more investment into science, and there's any talk about now we really learned our lesson. Now let's put more money into science. I don't understand why that is not the case, but every time I think about it, how much money goes into building roads. Nobody would even notice if they built a few less kilometer roads every year and put all that money into science. That the science would benefit. The budget of the science would double. It's just the scale is ridiculous. How different it is, but yet, this doesn't happen. And I'm trying to do my what I can to improve this situation, but it's obviously a problem which is going to take some time, I think, to resolve. And obviously, some countries are doing better than others, but at the end of the day, I think it's a global problem. We're just not investing enough into science.
Professor Murn, do you have anything to add?
Yeah, I've sort of, I did experience quite a bit of doing science in Slovenia, or at least trying to, right? And, I can attest to how difficult things are when I compare it to conditions here, right? At least most places I've worked in the US. I don't know how things may have changed now, right? Because I've been out for a long time, so I'm not quite on top of things. But I do recall, how difficult often it was to get things moving, right? Because of very different reasons, right? For instance, right, if there's a directive, right, that certain type of research can be done at a certain, say, department or faculty, as we call it, faculteta, right? Then, you're sort of forced to kind of go with that, right? Not to sort of do more open-ended research, but you're sort of directed into a certain directly right way, because this is sort of the command from it's kind of hard to define where it's coming from, right? But it's probably driven by in large part by industry when you're talking biology, right, in many cases, right, and so forth. So I think that sort of blocking the progress and also the interest, right, by people who are trying to do good science. So, I feel that no matter where you are, or or what you're doing, the fact is that the funding of science, so it needs to be there, right? Science needs to be supported, and that support, if you drill deep into where it's coming from, it's going to come down to politics, right? And the end of the day, there's going to be some decision made by politicians to invest into science or not, right? And I think in Slovenia, but it doesn't pertain just to Slovenia. There are many other countries, right, that suffer from it to different extent. The problem is that when we're talking politics, and I don't want to talk about it, right, at all. I want to talk about science, but we need to be aware of that. We need to be cognizant of that, right? That politicians are aware mainly of their re-election cycles, right? And that's sort of a very short-term looking at things, right? So in that regard, right? So they will invest into things that yield immediate return within a couple of years that this re-election cycle, right, lasts. And so anything beyond that may be considered or viewed as sort of waste of money, right? Waste of resources. And so we really need to think beyond that. And I don't see any other way but to increase the scientific literacy of the public, like I said before, because only in that manner will people know who to elect, what to invest into. Long-term investment is critical in science, because there's very little immediate return, right? That's why there is so much of this translational research, right? That sort of is where people are doing incremental steps to move something, right? But in fact, there is very little little thinking or sort of this global view as to how things would progress in this manner. So I think this longer view of investment in science, I think that's something that could definitely benefit, right? Research in Slovenia, right? That could I think move things along. But again, there needs to be. It's very hard. It's very sort of almost easy to say things, right? But to get things started, I think that's a whole other story, right? And I think that ASEF and its mission, I think I'm a huge fan of it, right? And I support it very much, because I think that's a first step, right?
Definitely, yes, I completely agree. So at this point, I would like to thank you both for such a great conversation. And before we close off, are there any resources that you would like to recommend to our listeners that would be further interested in the topics that we discussed today? Maybe something like a book, an article, maybe a podcast, anything that comes to mind.
I started reading books recently. I didn't read books before, but now I started reading books related to science, but not necessarily, hardcore, hard to understand material. And one book that I really liked. So first of all, I would recommend, whoever hasn't read it, to read the What is Life by Erwin Schrödinger, because that's where biology is coming from. But more recent books. So there's one that I really like, came out just last year. It was written by a psychiatrist and a neuroscientist, Carl Dieseroth at Stanford. He wrote the book called Projections, and it's really sort of it delves into human cognition and emotions, and how those could be now disentangled and better understood through these methods that he heavily contributed to, such as optogenetics, for instance, right? That now allows you to understand emotions or feelings or conditions such as anxiety. You can really break this down into, let's say, increased heart rate, looking for, avoidance behavior and so forth. So you can really break down these complex emotions by using technology. And this book is really beautiful because it sort of, portrays that in a very, very, easy to understand manner. and it makes you aware of what's around there. And books like that, a couple of them are now around coming out. I have a list that I could probably recommend, but that's one that really got my attention, really, and I couldn't put it down, really.
Yeah, like Jernej, I also started to read books more. I think there was a period where I, just in work, I had to read so much that I kind of didn't really find time anymore to read books so much. But I got back to that, and one suggestion, which is I guess a pretty boring suggestion that I would have, but it's a book that I read now a few years ago, and it really changed how I view things, is Yuval Harari's *Sapiens*, because it really kind of put things, put a lot of like very big picture things in place for me. Just, a lot of connections I never thought about, how really these things fit together. I mean, it really a lot of things started to make much more sense after reading that book. And another thing that I started kind of reading is books about, biographies about scientists, because I think it tells you a lot about how science is done, how it can be done better. What's the basis of that discrimination we hear about, in terms of gender and everything else. So one I recently read was, a book about Dorothy Hodgkin, Crowfoot Hodgkin. So she's one of the early crystallographers in kind of working on more complicated molecules, including macromolecules like proteins, sort of in the middle of last century. A Nobel Prize winner. So she actually wrote a little autobiography, which I haven't read, but this is a biography written afterwards by Georgina Ferry, but much more kind of looks like much more comprehensive, and I think very good, gives a very good insight in how things were done, what were the obstacles that researchers faced or scientists faced at that point in time, many of which I think extend to nowadays as well. But yeah, I've really enjoyed reading her life story.
Okay, thank you very much, both of you, for such a lovely conversation and book recommendations. And to our audience, thank you all very much for joining us for this podcast. We will be glad to hear your feedback, and you can send us an email to podcast@asef.net. Please follow ASEF on social media to learn more about our professors, fellows, and many more activities hosted by ASEF. We hope to see you in our next episode.